very good morning, ladies and gentlemen. Welcome to Breakfast at UMHealth webinar series. I would like to introduce our first speaker today, Dr. Farah Aida binti Dahalan from Imperial College London. Dr. Farah obtained her bachelor degree and master's in biochemistry from Universiti Malaysia, and she is fascinated with infectious disease research, particularly in malaria. And she graduated from Imperial College London in 2017. Currently, she's doing her postdoc research in Baum's labs in Imperial College. Her current work involves sporozoid, which is the infectious form of the human malaria parasites. Dr. Farah, welcome for your talk on a single-cell atlas of Plasmodium falciparum transmission through the mosquito.
Thank you, Prof. Lau. Hello, everyone. Assalamualaikum. I'm Farah. As Dr. Lau says, I'm currently working in Imperial College London. I'm back for holiday now, so I just thought of doing some sharing with all of you on the latest technology that we are working on, which is the single-cell sequencing. So this project. We had just published this paper recently in NatureCom, and I think it might be interesting for some of you in this talk. So I know that the audience consists of many people from different backgrounds, and it's a very wide audience. So I'm going to walk you through some background of malaria. And as you know, malaria is one of the oldest parasitic diseases in the world, and it's been there since 2.5 million to 30 million ago, and it got transmitted to America in the middle of 1500 to 1800. So there was one time where in the world, there are many countries with malaria, but due to some control and also prevention taken, some of the countries has malaria zero, where there's no transmission happening at all, such as in Europe countries and also in America and Australia. However, this transmission of malaria is still ongoing and causes a lot of death, especially in Africa region. And as you can see in the map. And last year, due to pandemic, the cases has increased from the year before. And so this is a bit worrying because, as you know, in Africa, the poverty level is very high. With so many other diseases, malaria is definitely not helping or not reducing until now. So the malaria control, prevention and treatment, despite that it's being one of the oldest diseases in the world, we can separate them into three different areas. The first one, which is the vector, which is targeting the mosquito. The type of prevention or control for vector is the use of bed nets. So these bed nets are embedded with the insecticides. They also spray insecticides in the house or outside of the house. And another way is to control the vector is by producing transgenic mosquitoes, such as CRISPR, using the CRISPR technology. So there's one group in Imperial who's producing transgenic mosquito that is able to only produce male progeny because only female bites, right? So that's from the vector perspective. And then the second one is the use of anti-malarials, which are the drug. And I think this is the much more common that people have seen or people have heard before. So there are two drugs that are currently being used or actively being used, which is the chloroquine and also artemisinin, or the combination of artemisinin. And the third one is the vaccine. And as you know, recently WHO recommended the use of this vaccine, which is called RTS vaccine. However, their downfall for all these three prevention, as there are so many insecticides resistance being reported, especially in Africa. And then recently in Nature, they showed that the only anti-malaria that is working in Africa has also shown a drug resistance, which is very worrisome. And the next thing is that, as you know, the vaccine for malaria, the efficiency is very low, and the protection over time reduces. It doesn't stay long. So basically, the memory T cells didn't remember that very well. And hence that's why malaria research is still ongoing, and there are more area to be ventured into in looking for new anti-malaria targets and vaccine development. So in this talk, I'll be talking about Plasmodium falciparum, which is one of the deadliest cause of malaria in human. As you know, malaria can infect like different species. So for malaria transmission to happen, it needs two hosts. One is the human to complete a sexual cycle. Basically, what happens is that they invade the red blood cell, they grow in the red blood cell, and it bursts and releases the parasite to invade new set of blood. And some of these parasites will commit into a gametocyte, which is the female and also the male gametocytes. And this gametocyte stays in the body. And once the female mosquito Anopheles bite the infected person, and if it's able to ingest gametocytes, this male and female will fuse in the midgut of the mosquito and then form ookinete. And this ookinete will embed into the mosquito epithelium cell of the midgut and develop further. This is where they produce thousands of merozoites in each oocyst. I'll show you what it looks like. And then over the time, like maybe around 2 weeks, the sporozoites are ready to infect the next person. So basically what happens is that it's burst from the oocyst, and the sporozoites goes to the hemolymph. Hemolymph is like the blood of the mosquito, and then it migrated to the salivary gland and ready to infect the next person when this mosquito takes in blood. Okay, so basically, the Malaria Cell Atlas is an ongoing project led by the group in Sanger Institute in Cambridge. So what they did was they take samples. So for this paper, they used the Plasmodium burgii, which is the Plasmodium that infects rodent, and then they take from every stage, and then they send for single cell sequencing. They got the data, they analyze the data, and then they have it in an atlas form, which is an interactive web. So you can go in and you can click for information. So what we did next is we expanded this atlas, focusing much more on the Plasmodium falciparum, which is the cause for malaria in human, focusing much more on the transmission, which is what's happening in the mosquito, particularly in the sporozoites perspective. Why we chose the single cell genomics as you can see is because when you have like the whole tissue, as you can imagine, and when you send them the whole tissue for bulk RNA input for transcriptomics analysis, you will get average gene expression for all of the cell. But when you do single cell sequencing, you separated each cell and then you send it for sequencing, you get the transcriptomics or gene expression per cell. So you can get like heterogeneity, you can reveal the heterogeneity between cells, and you get like different population, which is much more beneficial but much more informative. So I think only Malaysian can understand. If I can explain this in a, imagine ABC candle, you have like everything mashed up in the same bowl. You can see the colors, you can see what's there because we are Malaysian. But using single cell, what we can do is that we can decipher what's in there, not only like jagung or susu sejat or kacang, but you can also know how much of jagung, how many jagung, how many kacang. So I think single cell sequencing is really cool technology. Yes. So now I'm going to try to explain what are the things that we did before we go to the analysis. For sample preparation, we cultured the Plasmodium falciparum gametocyte around 14 days at 37 degrees in the incubator. And then once the gametocytes were formed, we fed them to the mosquitoes using a standard membrane feeding assay. And we isolate ookinete at day 1 or day 2 post-infection. And then usually on day 8 to 10, we look for the infection rate. We dissected the mosquito and try to see if they are infected and how many infection. And as you can see here, you can see these red blobs here. These are all the oocysts. So the sampling were done on day 12, day 14, day 17 to day 20. And at these different days, it represents different localization of the parasite and also different stages of the parasite as well. So all of these sporozoites were next sent for sporozoite isolation using the high gradient Q-dense to separate the mosquito debris and also lipid from the samples. And these samples were stained with drag 5 to stain the nucleus and CSP, which is one of the protein on the surface of the sporozoites. So they were sent for sorting using the flow cytometer machine, and the sporozoites were then captured in single 96 well plate, which has some buffer. And the first time when we did this, we got like 2 population, and we chose the population which has sporozoites by doing some simple qPCR, just to make sure that we have that population with sporozoites in there. So once we got that, the next thing that we did was send these plates for sequencing using a Hi6 25,000 machine, and we got the data, and we use the tools that is available to analyze the data. So this technology that we use for single cell sequencing is known as SmartSeq 2. Okay, so there are many ways to analyze single cell sequencing. I'm going to show you what we did in our paper to understand more about the biology of sporozoites during transmission. So the first thing that we did was we tried to look for the pattern of the gene usage across Plasmodium falciparum life cycle. And as you remember, our sampling are just done for the transmission stage. So what we did was we took the data from another paper, which is Reed et al. Paper from 2018, and they have the asexual stages, which consists of 161 single cells. And then we integrate this data together, and we produce this UMAP. So as you can see, the UMAP here, the reason of this doing this is that we want to make sure that the trajectory of the life cycle is there. As you can see, it's quite clear. Like the sexual cycle here, and then you can see the formation of the gametocytes and kinase. Next is oocyst, and then you can see like a mixture of hemolymph, injected salivary gland, and also activated sporozoites in this block right here. So this is really nice to see like the trajectory of the development of the parasite. And the next what we did was we find for the highly variable genes, and I'm going to use the abbreviation of HBGs. So from this, we produce k-nearest neighbors. So KNN graph is unsupervised. It's a supervised categorical analysis that you can use online to separate or to cluster the genes according to the function. So what is important from this is that it can predict gene function, especially for those genes which with unknown function. For example, if you look on the right here, when we do this clustering, we found that the sporozoites are highly expressed in G13 modules, and G13 modules are the cell surface. And that's interesting because when you want to produce, or when you want to produce vaccine, you need to know what are the surface proteins on the parasite. All right. The next, what we did was we integrate our data with the paper, the malaria cell atlas paper, which consists of Plasmodium berghei data set, and then we find for the conserved and diverged patterns of gene expression between these two species. And as you can see that they overlap nicely, the cell identity correlation is high. However, there's this one cluster down here, which is cluster C1, which has a much more data set from Plasmodium falciparum, and it is actually the oocyst sporozoites and also the hemolymph sporozoites. So because we want to compare it with the known data set, we remove this from the downstream analysis for differentially expressed genes. And as you can see here, once we have the data, we look for a specific gene in every cluster, just to make sure that it is representing different stages of the parasite. And we also look for the top 5 conserved marker genes between these 2 species. And in this heat map F here, we look for ortholog that were found to be highly expressed in PF compared to PB, Plasmodium falciparum compared to Plasmodium burgii. So what is important from this data set is that we know that around 30 to 40% of the genes, they have no function, no known function. So by having this type of analysis, we can make prediction about the gene function by surveying the correlation between the gene expression patterns along a highly resolved developmental trajectories. Okay, next, what we did was we recluster the data set, and then we align them to oriented them along a pseudo-time trajectory. So when we did that, we found a clear separation of these 3 clusters. And when you look at them from the stage of the parasites, you can see like a clear differential trajectories from the oocyst sporozoites to hemolymph sporozoites to the salivary gland and also injected sporozoites. And then when you look at on the pseudo-time trajectory, we can see that there's a mixture of sporozoites. So it's really heterogeneous. So it's not like sporozoites just were just in the same stage throughout their cycle. So basically their transcriptome changes over time and also over different location. Okay, the highly variable genes that we found highlighted that these sporozoites population are different. So they don't have any co-regulation between these genes. And this plot here kindly explain what I'm trying to share with you about the heterogeneity of the sporozoites population. Okay, next we chose one of the gene set, which is known as the Ep.50 transcription factor. So this transcription factor, they are important in driving major developmental switches during parasite life cycle. And we found just in sporozoites stage itself, they have different set of expression. Like some of the transcription factor is expressed during early stage of the sporozoites, and then it's being shut down at the later stage, and then some are expressed at the later stage and shut down in the early stage of the development across the sporozoites development. And the next thing that we found was the changes in the gene expression upon change in the environment. So for this particular question, what we did was we tried to imitate the transfer of sporozoites from mosquito to human. So we did like this experiment where we have a dermal fibroblast monolayer, and then we drop the sporozoites in the well, which consists of medium, and we collected the sporozoites that traverse through the dermal fibroblast. So it's kind of like mimicking the sporozoites goes into our skin. And then we collected the sporozoites, sent them for sequencing. We found that just using the medium is sufficient to induce the gene expression. And you can see the differences in the gene expression after an hour of activation compared to inactivated sporozoites. So what is important about this that we found, we further analyzed a particular gene, which is known as Ep.50. So this protein has never been studied in sporozoites before. P-Tax150 is a component of Plasmodium translocon exported protein complex, and it is responsible for protein trafficking into the host cell following merozoite invasion. So basically, this protein was mainly studied in the asexual stage, which is the merozoite invasion in the human. And then what we found was that this P-Tax150, they were activated after an hour. So we found that. We found that P-Tax has also been reported to be an adapter of a gene called ESP2 and HSP101. And ESP2 has been shown before, and that is necessary for sporozoite invasion. So this type of finding kind of like, lead us to hypothesize or like predict maybe P-Tax150 might have an early role in liver stage biology. So this kind of results can predict, the predicted results can let you plan for like further or that downstream analysis that focus on this particular gene. And on the right here are the expression that we found, and we showed that you can see the P-Tax expression after 1 hour and 2 hours of incubation or activation. All right. So basically, what I want to share here is that about the single cell sequencing and what we got from single cell sequencing of sporozoites. Basically, we can see or we can explore the individual transcriptome of every cell, every sporozoites, and we can explore the correlated gene expression trajectories, especially for those that has no known function. And we can also look for highly variable expression across cells of the same stage, and we can find, like differential phenotypes within the same population. And another thing that I want to share with you is the use of the malaria cell atlas, that especially if you're working with malaria. So Malaria Cell Atlas is an online interactive web tools which is free, and you can use which will aid in drug development and also vaccine intervention. So I'm just going to show you a little bit of Malaria Cell Atlas that you can go online, as you see here, sanger.ac.uk/tools. And they have the Smart Stick tool technology, and you can look at the UMAP clustering of the cells. You can look at the KNN gene graph, which shows the highly variable genes. And so these are colored by, you can choose by stage. You can choose different stage to understand how the expression is in every stage. And you can also select by genes. So this is how it looks like. And another way is for single cell sequencing is the Chromium 10X, which is what we are working on now. You can also get the data from Plasmodium burgeri, which is the rodent malaria. You can look at the stage and also the gene expression by gene number. So it's pretty straightforward. So I guess that by showing this, it might help you, especially if you're interested in malaria, to look for specific genes that you're interested in. And they have also data from the Chromium 10X. And they also added in data for Plasmodium knowledge. Okay. And if you're interested in playing with the data, because there's a lot of tools out there that you can download and try to, if you want to practice how to analyze the data, there's a lot of tools, and you can download the data, the raw data from this particular project code. Oops. Okay, so these are the team that we work together on. So from Imperial College London is, so that's Jake. and Eliana. So Eliana, me, and Jeannie from Sanger, we are the first co-authors for this paper, and this project is being looked by Jake and Mara from Sanger. So, thank you.
Thank you, Farah, for a very interesting talk. Actually, single-cell sequencing is getting very popular now in malaria research. Right, we are also interested to work on P-neuroscience in the future. So hopefully, by the time you are in Malaysia, you can help us out, bring back the technology from Imperial to Malaysia. We have a few questions. I also have a few questions, but I guess due to time limitation, I think we go to the chat box first. There's one question from Dr. Lee Wing Chow. He's asking, hi Farah, from my understanding, this work was conducted with sporozoites harvested from Anopheles stevensii. If the same parasite were to be inoculated into a different species of Anopheline, can we pick up the gene with altered expressions by the sporozoites in these different mosquito? What do you think if using different Anopheles mosquito?
Okay, so it's Wen Chao. Okay.
Yes.
I can't really comment about that because we have not done the same sporozoites on different Anopheles species, but I assume it might be the same. But maybe the difference might be from the host or from the vector transcriptome. But that's a very interesting thing. So one of the things that we are trying to do now is we're trying to collect samples from the field, and I'm trying to see if we can get a good transcriptomics out of frozen samples. That's what we are working now, and then definitely if that works, then we can conclude whether different species might give different transcriptome expression or similar. But that's a very good question.
Yeah, I thought so. So I mean, using different thing, I think should produce a bit slightly different results, right?
Yeah, but it can't differ so much because sporozoites.
Yeah. Right. Any limitation in using this single cell sequencing?
So basically, so basically single cell sequencing, because it is using RNA. So I guess that one of the limitations that we realize now is to get the samples from the field, and then you want to bring them back for sequencing. You can't have the machine in, for example, if you're looking at Africa and you're trying to see if you can sequence the parasite there, right? You can't have the whole machine there because it's very expensive, and there's a lot of limitation of the resources and also the machine. So I think that's one of the limitations. One more is, I think single cell sequencing is a bit pricey, despite the generation of the data, which is so big. So that's the limitation that I can see. Another thing is that I would say that the way of analyzing, that is also kind of a limitation, but there's a lot of things online now that can help you or aid you in trying to analyze. I'm not a bioinformatician, but it's quite easy to follow. It's just a little bit hard in a first time of analyzing because we're not bioinformatician, to understand this coding and all these things. So that's one of the limitations that I can see. But you can also do collaboration with some bioinformatics lab, which is easier.
Sure. So, okay, we have another two questions, more general question for Amir. So first question, he asked whether malaria can be prevented using anti-malaria drugs or vaccine. Second question, she's asking why male are getting more infections compared to female.
Who getting more infections? Male? The male people? Oh, okay. So the first question about whether the vaccine and drug is still working. So basically, because malaria is prevalent in Africa, right? It has been reported that there are resistancy. It's still working, but from what I understood is that because we have collaborators in Africa as well, it is starting to slow down. So basically, it's not really working well, and that's because of the resistancy. And for the vaccine is efficiency is very low, but WHO recommended it because especially it is affecting children less than 5 years old, and it's worrisome because they can die. So that's why they, like rather than not giving the vaccine at all, they gave the vaccine. So basically, I feel that it's not working very well, but it is at least preventing malaria from getting worse. So that's what I feel. And the second thing, why is it affecting male? I'm not so sure about that because I thought that it's the same. And I think malaria is affecting children more than the adults.
Right. I think Amir is asking in Malaysia. I think mostly on north side. We think male is more highest because outside is usually outdoor in jungle. So one of the prediction could be male. More activities for male happening in the jungle compared to female. Okay, so many questions. I think we can only take the last one because it's already 8:30. The last one from Dr. Nurshamimi about whether vaccine still work if you're already infected.
Vaccine is basically to prevent this is what I feel. If you're already infected and then you take the vaccine for malaria, I can't say that it's not going to work because there's no report on this yet. But I'm not so sure about that. I think we need to see how does the rollout of the vaccine reports. I think sure they will write a paper about this because it's a new thing. So let's see.
Yeah, we'll see.
Yeah.
We are now still looking for a good vaccine for malaria.
Exactly.
Right.
Yeah.
All right. Thank you. Thanks, Farah. We hope to see you again in our department. I can arrange for you after this.
Of course. I mean, anything can email me as well. I think my email is there.
Yeah.
I'm more than happy to chat. It's happy to be here. Thank you.
Thank you so much. I pass over the floor to Dr. Azlina. Thank you, Dr. Farah.
Thank you.
Thank you, Prof. Lau. Assalamualaikum and good morning. Hi, everyone. I'm Azlina from the Department of Molecular Medicine, and I will be moderating the second session. So our next presenter is Dr. Shamimi Rashid. Dr. Shamimi is an associate professor in the Department of Molecular Medicine. She is a alumni, having obtained her undergraduate and master's degree here. She comes from a background of genetics and molecular biology. Dr. Shamimi also did her PhD in Imperial, London, under the Malaysia Imperial doctoral program. Her main research interest is in the field of cancer research and virology. She was recently awarded the Gold Medal and Special Prize for her invention on skin anti-aging from local plants at the Korea International Women's Invention Exposition. And she will be sharing about this research project in her talk today, entitled Antioxidant and Anti-Aging Potentials of Local Plant Extracts in Normal Human Dermal Fibroblast Cells. Off to you, Dr. Shamimi.
Right, thank you so much, Prof, for a kind introduction. Assalamualaikum and very good morning to all. Okay, so my name is Nurshamimi Rashid. I'm from Department of Molecular Medicine, Faculty of Medicine UM. So the title of my talk today is Antioxidant and Anti-Aging Potential of Local Plant Extracts in Normal Human Dermal Fibroblast Cells. So I think my talk is very much simple than Dr. Farah. So I'm going to share to you guys regarding what we have done for this work. Okay, so just a little bit introduction about skin aging, because this work is we want to look into the anti-aging effect of the local plants extract on skin. So skin aging is a complex biological process influenced by a combination of intrinsic and extrinsic factors, which lead to cumulative alteration of skin structure, function, and appearance. Okay, so there are two types of skin aging: intrinsic aging, which is the aging caused by the passage of time. So this is the inevitable physiological process. Okay, such as you have a fine wrinkles, sagging skin, and some pigmented spots. And then another one is the extrinsic aging. This is the premature aging due to the environmental factors. Okay, where you have a deep wrinkle, leathery appearance, and motile pigmentation. Okay, and this figure here show the symptoms of intrinsic and extrinsic aging. Okay, on human skin. So basically, if we expose more to the environmental factors, then our skin will get much more age rather than it should be. Okay. All right. So just a brief introduction about the pathway of skin aging. So for the skin aging, right? So mainly here the UV radiation will induce the ROS production, in our skin, and then with cumulative of ROS production, then it will increase this enzyme here, hyaluronidase and elastase, which then makes this hyaluronic acid and elastin reduce, and then it reduces in the collagen production in our skin, and also higher production of ROS leads to the higher production of these transition factors, AP1 and the MMP enzyme, which also leads to the reduced production of collagen, which then you have the aging symptom of your skin. Okay, so that's why for this work, right? Because we know that some of the people out there, they cannot use cannot use the cosmetic with a chemical inside the cosmetic, because they have some of them, they have some allergic reaction. So with this, the hypersensitivity skin, they are much more suitable for the nature product cosmetic. So it is necessary for us to seek out for safe and effective skincare cosmetic from the natural resources. So that's why we're using we screen the local plants extract from Malaysia. Okay, and then the secondary metabolites of plants are phytochemicals, which exhibit antioxidant activities and possess a beneficial physiological effect. So example of secondary metabolite, the first one is flavonoids and the allied phenolic and polyphenolic compounds. And the second one is terpenoids, and the third one is nitrogen-containing alkaloids and sulfur-containing compounds. So this is an example of the beneficial effect of aloe vera and green tea. Yeah, it's very famous now, that widely studied, and I think most of us we drink a green tea as our cup of coffee every day, and which it has a high antioxidant activity. Okay, so when we talk about the aging of the skin, so we want to first we want to inhibit the enzyme, and then we talk about the antioxidant and also the current approaches in anti-aging studies. Okay, so for the first we talk about the enzyme inhibitor. So the enzyme which involve MMP and hyaluronidase, elastase, and some other enzyme as well. So we inhibit this enzyme function, and then the structure integrity of the skin. By inhibiting this enzyme, then the structure of the skin integrity is preserved, and therefore the appearance and the formation of aging symptoms will be reduced. Okay, and secondly, the antioxidant. So antioxidants play a big role in free radical scavenging and neutralizing to indirectly diminish the ROS production. Okay, so our current approach for anti-aging study is several plants. We know that many plants is actually have a potent antioxidant and enzyme inhibitors and enzyme inhibitor for this properties, which associated with reduced incidence of ROS and photoaging. Okay, so where are we at the moment? Yeah, formulation. Where are we at the moment for this local plants development for in terms of medicine, in terms of cosmetic and other things. So herbs or local plants have been identified as a new source of economic growth in Malaysia since 2011. And we already know that our country, we are rich with flora and fauna. Okay, so we have more than 4,000 or 5,000 of flora species in our country, which are yet to be studied. In terms of alternative medicine, in terms of to develop as a drug, or in terms for cosmetic studies. So since then, since 2011, Ministry of Agriculture, they have started with the Entry Point Project 1. Okay, so under this project, they have found out 18 herbs and 10 other species which will be developed towards the for alternative medicine and also for the drug development. Okay, so at the moment in Asia, China dominated the herbs industry, which is worth USD 48 billion. Okay, so this is big money here. So our country, Malaysia, is one of the largest contributor to the world's biodiversity, and we are ranked number 12 in the world and number 4 in Asia as diversified country. So as like I said just now, like I mentioned just now, that we have more than 5,000 species of plants in our country. Okay, so we are very rich with that. So in Malaysia, herbs have been classified by product groups as follows. Okay, the first one, we classify into flavors and fragrances, which can be produced for cosmetics, perfume, oil for aroma, essential oil, which I think now essential oil. I think some of us we use essential oil. Okay, to reduce stress. And some other things as well. And then beverages. Okay, secondly is in terms of pharmaceuticals and herbal. This one to develop alternative medicine, drugs, or any types of vitamins and supplements. This is what Marty's been doing at the moment. Okay, where they come up with the black ginger coffee or tea, I think. Yeah. So, and then the third one is in terms of health and functional food. This one in herbal teas, like we have the green tea. We also now we have a green coffee, which they said that it can help assist you to reduce your weight, and herbal supplement. And lastly, biopesticides. Yeah, this is like for insect repellent, crops pesticides, and household pesticides. Okay, this one just want to share with you. This is the World Herbal Market by product category. Yeah, I'm sorry, it's not really clear. Okay, but the blue one, this is the Western herbs. Yeah, which comprise of 45.9%. Okay, which is a lot, and then followed by the Chinese herbs. Okay, 35.5%, and then this is a homeopathy and the Ayurveda. yeah, the Indian traditional medicine here. So as you can see from this map here, the Western country, they are using these herbs in a lot of things, yeah. And then followed by the Chinese people, yeah, because Chinese, they have this Chinese alternative medicine. So that's why, you know, China dominate the herbs industry in Asia. All right. Okay, so the problem statement for this. Okay, so as you all know that most of the commercialized skin anti-aging regime use plants originated from Western countries. Yeah, if you, as you already know that most of the cosmetic company like Estee Lauder, Lancome, or some other things, they use their plants. Okay, Clarins, they use the plants from France. They use a seaweed from their ocean. So I think now it is our time to try to investigate the whether that, you know, our plant, we have a potential to reduce or stop the aging for our skin. Okay, so that we can develop for the cosmetic purposes. Right. So secondly, the work of Malaysian anti-aging studies has been very limited and lack of continuing development. And then to assist people with allergic reaction, okay, because natural-based product is more suitable for hypersensitive skin. And then by utilizing our local plants as a skin anti-aging regimen, which then may assist in the development of our agriculture in Malaysia, and then to answer the Ep.50 project, okay, by the Malaysian government, because now I think government, they put quite a huge amount of money for the herbs or local plants development for a lot of things. Okay, so we have this is the list of the plants, local plants, yeah, Malaysian plants that we use for this study. We have 30 in total, and most of these plants we use their leaves to extract, and some of it we use the seed pod, and like bunga lawang, we use the fruit. Okay, and so we screened all these 30 plants. Okay, so the first one, okay, so I divide the work into 3 parts. So the first part is the screening of local plants with potential antioxidant and anti-aging activities. So we have 30 selected plants extract. So like the list just now. Okay, so we screen for the phytochemical constituents in terms of the total of phenolic and flavonoid content, and then followed by 15 selected plant extract. We test for the antioxidant, anti-aging and cytotoxicity study. This is what we did, DPPH and ABTS, and then we look for the inhibition of the collagenase elastase and hyaluronidase enzyme, and this is the MTT assay to look for the toxicity of the plant extracts in the cells. Okay, and from this, we select 4 plant extract. We test more for the antioxidant, anti-aging and cytotoxicity with the same test. And after that, we identify and screen the plant extract compound using HPLC. Okay, so from this, from part 1 study, we know that different plant extracts have different capability as antioxidant and anti-aging. And the capability of the plant extract is depending on many factors, such as the extraction method, how you extract the plants, sorry, and then the individual compounds in the plant, and then the plant parts used for the extraction, and the condition of the cultivation of the plant. Okay, so all of these it plays a role as a capability for the plant, for the plant extracts. So from the part 1, okay, so we we can say that this Patobeetle, Catechu aristatus, and Bosenbergia rotunda, and these 4 respective fractions are recommended as the candidates to obtain efficient antioxidant and anti-aging compounds. Okay, so following this, we have a part 2. I cannot see that. Okay, our part 2 is the investigation on the effects of the antioxidant activities of selected compounds on ultraviolet B induced aging in normal human dermal fibroblast cells. Okay, so for this part, we use 15 compounds from local plants. Okay, and then following this, then out of 15 compounds, we select 6 compounds at a different concentration. And for this one, the cells that we use, the NHDF cells, we expose the UV, and then this is how the photoaging will be induced. Okay, is induced to the cells, and then after that we apply the compound treatments, okay, for the cells, and after that we measure for the total ROS and the this antioxidant enzyme assay. And then following this, we also test for the expression of the gene, okay, which related to the antioxidant defense using the qPCR. All right, so for second part, in terms of a result and discussion, okay, so the result demonstrate, for the second part, the result demonstrate that these compounds act at different stages of antioxidant mechanism, like scavenging the ROS, stimulating the antioxidant activity, and manipulating the protein and gene related to antioxidant defense in normal and UV induced NHDF cells. Okay, so despite many studies in regards to the antioxidant effect of allergic acid and eugenol, okay, so these are the 2 compounds that we found which works really well for the antioxidant and anti-aging. So the protective effect of this compound against oxidative damage to normal and UVB induced and HDF cells has not been extensively investigated. Okay, so therefore our findings in this study, even though it's limited to the in vitro model, so from this part we may suggest that this allergic acid and eugenol have a beneficial effect in protecting normal and UVB induced NHDF cells against oxidative damage. Okay, so following this, we have the third part of the study to investigate the anti-aging properties of selected compounds on ultraviolet B-induced aging in NHDF cells. Okay, so for this, we use 15 compounds of plant. Okay, and then after that, we select 6 compounds from this. At a different concentration, and then we induce the cells with UVB, induce the cells to aging with UVB exposure, and then after that we treat the cells with the compound, the 6 compounds, and then we measure for the total collagen and the aminoglycan content. And we also measure the expression of the gene and protein related to the aging process using PCR and the Western blot. Okay, so for this, what we found is this result demonstrate that at various concentration, the quercetin, galangin, and eugenol have a better anti-aging effect compared to other compounds when tested on the normal and UVB induced NHDF cells. So even though many researches were carried out on plant for anti-aging properties, the study for anti-aging effect for specific compound found in plant is still limited. Okay, so basically most of the paper that when we read, they carry out on the plant extracts, but they don't really go further deep to find out what are the specific compound which actually works for the aging. Yeah. So therefore, the information obtained from this study will provide the basic data for the anti-aging effect on normal and UVB-induced NHDF cells of this compound, especially quercetin and eugenol. Okay. So for this work, right, we showed that the piper beetle, Aristatis catechu, Aristatis and beta boson beja rotunda and their respective fractions are recommended as the candidates to obtain efficient antioxidant and anti-aging compounds. So basically, from the 30 local plants that we screened, these 4 plants is the best for the antioxidant and anti-aging. Okay, so compound obtained from these plants, allergic acid. Okay, the first one is allergic acid, which have a good antioxidant capability. And then second one, quercetin and galangin, which exhibit better anti-aging activity compared to other compounds. And on the other hand, eugenol, which coming from Syria here, piper beetle, have the capability of both good antioxidant and good anti-aging activities. Okay, so these findings provide a reference and information on the mechanism of antioxidant and anti-aging. But there are also other mechanisms and parameters related to skin aging, like anti-inflammatory and cell senescence, which can be applied for further research. So now we want to look further in terms of a cell senescence, what this compound can do for cell senescence. Okay, the mechanism behind it. Okay, and then the present study has revealed the importance of this compound and plans to fight skin aging, and we can use this information for the development of inexpensive, safe, and effective anti-aging cosmetic. And this information can also be used to test the synergistic effect between this compound, between quercetin allergic or eugenol and quercetin, and which focus for the anti-aging properties. Okay, so this is the key findings. So the key findings from this study is this 4 plant: cat's whiskers, Chinese keys. Okay, so these are the respective compound: quercetin from Chinese keys, galangin from cat's whiskers, eugenol from piper beetle, and ellagic acid from pinang palm. Okay, which they have the potent radical scavenging activity, and they have high anti-aging properties. And they can also reduce the, they have a potential to reduce the intracellular ROS. But from these 4, eugenol showed, eugenol is the compound that showed both, which has the antioxidant, good antioxidant and good anti-aging activity. Okay, so for this work, we won a special prize and gold medal from this Korea International Women's Invention Exposition (KIWI), and under Korea Institute of Patent Information. Okay. This is just recently. Okay, and this work, we have worked closely with Mardi, and they are very excited about it. So because of the good activities of the compounds from our local plant extracts. Okay, and this company has also interested to work with us. So this is a company from the US because they are interested to collaborate with us, because this is like the first group, we're like the first group that showed that our local plants is actually we have better effect for anti-aging. Okay, so we have these 2 collaborators. Okay, so at the moment, this is our ongoing work because due to the funding limitation, we cannot proceed with the in vivo. So now we're doing the in vivo study for this work, and in future we want to look into the diabetic foot ulcer, because now we know that one of the methods to combat the foot ulcer that you can target on the cell senescence. So this is what we want to do. We want to use the compound, and we want to see whether it can stop the diabetic foot ulcer. Okay, so this has been patented. Okay, so these are all the publications and conferences from this study. Yeah. Okay, so I would like to thank you, the key person of this study. Yeah, Prof. Fatimah Ibrahim. She's from Biomedical Engineering Department, Universiti Malaya. Prof. Dr. Rana Yusof from our department, and then this is the student which carried out the project, Dr. Zaliatul Akmal Mat Yasin, and the grants that funded this project, UMRG and the postgraduate grants. So with that, I thank you.
Thank you very much, Dr. Shamimi, for the very beneficial talk. Certainly, we can see a lot of potential, for our natural products and to develop our herbs. Okay, just going through the chat box, Mimi, there's a question from Dr. Yu asking whether you are working with the Science and Agricultural Unit in UM to look at growing herbs and educating the local population to grow the herbs. That be something that you would plan on doing in the future? It's not already done.
Thank you for the question. Actually, we collaborate with Mardi, and at the moment, it's something what we plan to educate lay people regarding our plant extracts. Our local plants. Yeah, the potential of using our local plants. So that's what we want to do in the near future. Yep.
Yeah, good. Another question is, if you were to consider to use the herbs, for diabetic foot ulcer, what do you think would be the cost estimate for, let's say, maybe 2 to 3 weeks?
I'm sorry. Can you repeat the question?
If you are to use your herbs for diabetic foot ulcers, what would the cost estimate be like for a course of maybe 2 to 3 weeks?
Oh, okay. Cost estimate. Okay, so this is what we've been discussing about with Prof. Fatimah and Prof. Rana and our Mardi group as well. And this is for our plan, because at the moment we apply. We just sent out for the grant application for this project. So the cost estimation for about 2 to 3 weeks, it will be about around 10 ringgit. So that's what we estimate, but we don't know yet. So that's because for this work, actually, we really looking forward for it, and this is what we want to do, because we want it to be cheap, but it is effective. Because we know that most of the diabetic people, they come from various backgrounds. So we want to help people so that they can use this cheap, safe, but effective for them. So we have calculated this. It's about 10 to 15 ringgit for a month. Yeah.
All right. Okay. Thank you so much, Mimi. We are coming up to 9:05 already. So we thank you so much for your presentation, and we look forward to your team finding the magic bullet that can keep us all looking useful forever.
Thank you.
With that, we end this week's session of Breakfast at UMHealth. Thank you, everyone, for joining us, and wishing everyone a fruitful day. Thank you. Assalamualaikum and good day. Bye.